Hello everyone, my name is Tanushree Dheer from School of Sciences. In today's video, we will be discussing about diatoms. The major areas on which we will focus include basic detailing of diatoms, their extraction methods and forensic significance. So let's start. Diatoms are living algae. They are unicellular photosynthetic, either free-floating or immobile organisms found in nearly all types of water bodies. They also inhabit terrestrial habitats such as wet rock, mosses, and even caves. They occur as solitary or sometimes in colonies. Taxonomic classification includes division Chrysophyta, class Basilerophyceae, order Centrales, and Pinnales. Now, if we talk about the structure of diatom, then first comes their outer covering or cell wall. Diatoms have an outer siliceous skeleton called frustule. Each frustule is made up of two halves, which is known as valve or theca. One valve is slightly bigger than the other so that they can fit within each other well. The larger theca is usually thought of as upper and is thus termed as epitheca. The smaller theca is usually thought of as lower and is thus termed as hypotheca. As the diatoms divide, each daughter retains one of the theca of the original frustule and produce one new theca. The next important structural component is raphe. It is a slit-like structure extending lengthwise along the cell, allowing the diatom to glide along the surface. For movement, diatoms possess some additional secondary structures like bristles. If we talk about body symmetry, then centrales have radial symmetry and pinnales have bilateral symmetry. Now, on the screen, you can see two different species of diatom. Figure 1 represents centrales. Figure 2 represents pinnales. This central longitudinal section or division is known as raphe, and these structures present on the side of the diatom is known as striation. Both raphe and striation present in the diatom form the basis of their microscopic identification. Next is the extraction method. There are mainly four types of extraction methods: acid digestion method, enzymatic digestion method, ultrasonic radiation method, and use of strong ionic detergents for digestion of cell. Let us first understand acid digestion method. In this method, for digestion of biological material, strong acids like nitric acid and sulfuric acid are used. Firstly, 50 gram of tissue sample is taken and is suspended in concentrated nitric acid and kept in a fume hood for approximately 48 hours. The suspension is then subjected for centrifugation, 30 minutes at 2000 to 3000 rpm. The centrifugation is done 2 to 3 times. The supernatant is discarded and microscopic slide is prepared using the residue for diatom analysis and examination. The second method is enzymatic digestion method. In this method, cell digestion of biological evidence is carried out by using proteinase K and tris HCl buffer. 50 gram of tissue sample is taken and suspended in 500 ml of proteinase K and 0.01 molar tris HCl buffer. After digestion, we will follow the same process of centrifugation and slide preparation. In ultrasonic radiation extraction, low frequency and high power ultrasound waves are utilized for cell digestion. Lastly, in strong ionic detergent digestion method, detergents like sodium decyl sulfate and hydrogen peroxide are used. These detergents denature the protein and disrupt the membrane by breaking protein linkages. Now let's focus on the significance or importance of diatoms. First is determining antemortem and postmortem drowning. For understanding this, we need to first understand what is drowning. So, drowning can be defined as respiratory impairment when air is not able to enter the lungs due to submersion or immersion of mouth and nostrils into water or any other type of liquid. So, if the person is alive, before being drowned in water, the diatoms will penetrate into the alveolar system and bloodstream due to breathing in water by victim. This is known as antemortem drowning. But if the person was already dead before drowning, then there won't be any penetration of diatoms inside the body. This is known as postmortem drowning. The results of examination of diatom found inside the victim's body when compared with reference sample of water taken from crime scene helps in establishing post-mortem and ante-mortem drowning. The second significance is determination of crime scene or location. As diatoms are region specific, climate and temperature specific, thus examination of diatom species can help in establishing the scene of crime and time of crime commission. So this brings us to the end of our today's session. I hope you might have found it interesting. Thank you for watching.